faiths of the world. So that'll be an interesting little switch. This is Dr. Minarsik. Today is August 29th. It's 9 o'clock in the morning. This is our 9th webinar. The topic is immune diseases. And why am I saying yikes again? Well, I'm saying yikes again because the last webinar we had to cover genetics diseases, and it was such a big topic. I keep going, how could I do it in 2 hours? Well, we're going to give it the best shot, but it might be a long day because there's a lot of stuff. And, up until now, I've been keeping the webinars like significantly less than 2 hours, maybe hour and a half or more. But today we might go with a distance. Let's do our AVM test under the butterfly and make sure everybody's hearing and seeing well. A stands for audio. Question is, can you hear me well, and did you hear the music well? So I'm hoping for a nice answer there somewhere. Good, I got yeses. Second question is video. How many fingers do I have up? That way we'll know if the video and the live feed is working as well. And you got that one right, but guess what? I tricked you. It's not 5 fingers, it's 4 fingers and a thumb. Okay, the last question is, I tried to do something to improve the music today. Did the music, well, on a scale of 1 to 10, with 10 being super, what was the quality of the music today? I get 7s, 8s, 9s, and today I got 7s and 8s. Okay, well, maybe that didn't improve it too much, but it's still pretty darn good. Today, our topic is immune diseases. And once again, this is a massive topic, and I cringe when I do this. How can I do in 2 hours what maybe the students are taking an entire course in, or maybe took the entire course last year? So we're going to concentrate on the basics of immunity, more or less, and then go into the classical things that could go wrong with the immune system. How about if we pop open our PowerPoint and pop down the ugly guy, and we will be able to start our discussion of Chapter 6, Immune Diseases. There we go. Might as well start at the top. I thought a nice little say a few words about immune diseases in general. Now, I think you all have a good idea of the classical mechanisms of what we call immunity, whether they're acquired or innate. But I think over the years, the concept of immunity has been expanded quite a bit. We all learned about antibodies and T cells and stuff, but I think when you look at it, you might just have a wider definition of the immune system, and you could probably say it's not just things like antigen recognition and, killing of cells. It's basically all of the sum total of events and processes and cascades that keep the body from getting a disease. And now our concepts of immunity are really, really expanded into just about every disease there is. So just like we said in the last webinar, that all diseases are genetic diseases, I think we could probably say again, yikes! All diseases are immune diseases because a disease is something that has broken down your normal state of healthy well-being. Okay, so what we're going to talk about today are the basic concepts of immunity, the innate versus the adaptive. The innate is what you're born with, and the adaptive is what you learn on the basis of being exposed to certain antigens or microbes. We're going to understand the basic roles of all the basic cellular players, like the lymphocytes and the macrophages are the two, and they have a lot of derivations like dendritic cells, NK cells, and so forth. And we're going to once again, for the third time, look at the major cytokines directing a lot of the features of immunity, particularly the acquired immunity. And we're going to talk about something that's always has to be discussed, and it's in all the textbooks, and sometimes it doesn't make too sense logically, but there are basically 4 different ways, or you could say 4 different types of hypersensitivity, or 4 classical ways you might say the immune system might break down. we're going to have to go under this big understanding of the autoimmune diseases as well, okay? We're going to talk about two types of autoimmune diseases. We're going to talk about the so-called systemic, which involve the whole body, as well as the localized ones. But I want to tell you that, first of all, some of the things that we always considered localized diseases are now being looked at from a broader spectrum. And also, you could also say that just about any chronic inflammatory disease which is not known to have some type of infectious or microbial etiology, is very often now, over the years and over the generations, being lumped under the category of autoimmune diseases or immune recognition failure. Okay, we'll talk about some of the things that could go wrong with the players of the immune system, whether you're born with them, the so-called primary or genetic immunodeficiencies, or the secondary, the acquired immunodeficiencies. So if you say acquired immunodeficiency disease, you know that means AIDS, okay? That's one of the major acquired ones, of course. But then there's a whole big family of the primary or G. So you can say all immunodeficiencies are either AIDS or GIDS or PIDS, but that's just a crazy way of looking at it. And of course, we're going to go into the actual basic discussion of some of the immune mechanisms in AIDS, clinical, other types of features, and then talk about an immune type of disease that nobody ever will probably deal with directly, although I notice in the literature, amyloidosis has been becoming more popular in the role of vascular disease than it has been, because it's very common with aging, but just not as common as atherosclerosis. Okay. So immunity, what is immunity? Well, we said that immunity was a sum total of all the mechanisms in your body to protect or defend you from getting all kinds of diseases. Well, that's nice. But classically, we divide immunity into innate and adaptive. And innate immunity are the things that you're born with, okay? Adaptive immunity is an acquired immunity depending on your exposure to pathogens or antigens, or in a broader sense, antigens which are not recognized or tolerated by the MHC. Now, I know you're probably thinking, well, what is this big MHC? Well, we're going to be discussing it several times because it is the genetic or chromosomal origin of the sum total of your adaptive immunity. You could think of it that way. Chromosome 6, the big MHC place on chromosome 6. So specifically, you have antigens all over the surfaces of your cells, whether you're a mouse in the H2 system or a human in the HLA system. And those antigens are determined primarily by a part of chromosome 6, P, which of course is the short arm or petite. There's a certain area there that codes not only for most or all of the antigens on cellular surfaces, but for all of the mechanisms with immune cells and antibodies to recognize those or to tolerate those or non-tolerate those. And then there's another part, not just the antigens or the recognizing proteins, but also another part of the MHC is the complement system. And I know you're thinking, well, why the hell should the complement system be included in this mess? Well, don't forget, when an antigen attacks and when an antibody attacks the antigen on the cell surface, there is that whole system of complement proteins that kicks in if the surface of that cell is lysed or broken or ultimately killed, let's say. So what is innate immunity? Well, one of them is barriers. You know, we have skin, we have mucosa, we have anatomic barriers, we have lymphocytes and immune cells, often very close to those proximities of the barriers which separate us from the outside world filled with zillions of potentially nasty antigens and microbes. We have this whole population of soldiers and cells. You know, some of them are generals, some of them are sergeants. We're going to talk about the roles of the lymphocytes. the macrophages, which are the two main ones. And if you want to think of a plasma cell as being a specialized form of a B lymphocyte, you could do that. If you wanted to think of a dendritic cell as being kind of a specialized type of a macrophage because they come from the same line, you could do that. And if you want to think of the NK cells as soldiers by themselves, or ultimately probably derived from the lymphocytic series, you could do that as well. We're going to once again talk about the various cytokines' role in regulating the immune processes, the main one. We've already talked about the complement and the coagulation factors, but we'll show you how that fits in as well. Many of the plasma proteins. And finally, parts of the system that you're born with. Remember, all of these are not acquired. You're born with these. Are the receptors that you get on your macrophages, okay, called TLRs, okay, the toll-like receptors. And even though we said this is innate immunity, the toll-like receptors on macrophages somehow in evolutionary history have learned what the common bad guys are. So they don't have to be exposed to it. They are already born with it, okay? So many people think that because it's basically a receptive or a way to recognize cells, it would be recognize antigens that it's never been exposed to. That's kind of an interesting concept that is often not discussed in immunity. Adaptive immunity is the immunity, unlike the innate immunity that we adapt to. We're born with the tools for it, but we learn how to do it. So if you want to think as cellular immunity or cells which directly have reactions with bad or external antigens, you can do that. And if you want to think of humoral immunity or antibodies also as being sensitized by microbes or antigens and therefore learning them. So adaptive, I think you could probably say another good word for adaptive is learned, because in order for cells to react with bad guys or antibodies to react with bad guys, they have to be exposed to them first. They have to know who they are. And on the basis of that, they create mechanisms of learning to where they can now be more ready or sensitized for them. Technically, you build up antibodies after your first exposure to antigens which your body does not tolerate. Here's a general scheme again. I know some of it is in very small letters, and even I can't read some of them. I doubt if you could read every single thing here, but if you want to think of humoral immunity or antibody-based immunity, humors, as being something that is generated ultimately by the type of lymphocyte known as a B cell, or an even more specialized kind of a B cell called a plasma cell, which stores antibodies in great numbers, as on the basis of having learned these antigens, and sometimes through the mechanisms of T cells as well, coding these antigens with antibodies, okay, and then kicking in the complement system to lyse these cells, which are then ultimately phagocytized. So that's how you kill the bad guys, okay, from humoral immunity. You could probably regard that as more of a advanced type of immunity, because now the cell kind of keeps its hands clean and he just sends out bullets or arrows and he doesn't have to fight the bad guy hand to hand. On the other hand, in the cellular immunity, you have direct cellular contact. Classically, you have an antigen-presenting cell directly interacting with untolerated antigens or microbes, then sending those signals to a T cell, and if it's a helper T cell, the helper T cell will generate various cytokines, which we mentioned, okay, which will ultimately regulate more lymphocytes to get involved. And some of those lymphocytes, the so-called effector T cells, can then do a direct hand-to-hand killing of the infected cell, ultimately, once again, resulting in phagocytosis and complete destruction and cleanup of the battlefield, the micro-battlefield. So what are the cells of the immune system? Let's recognize them and let's know generally what they do. Well, you all have heard of T and B cells. You know, when this concept first came out, it was actually during my lifetime, believe it or not. We didn't think much of it. We said, oh, well, B cells and T cells stand for a very common English word called bullshit. Okay, because they don't really, why should you lump them into that? But it's the most important, probably, division of lymphocytes, is the concept of the cells which ultimately produce antibodies and ultimately the cells which either regulate or destroy the antigens directly without the use of antibodies. They call it T because the lymphocytes of the T system are developed in the thymus. And they call B cells B because they ultimately come from a type of tissue present in birds and not really seen too easily in humans, called the bursa of Fabricius. Or some people say, well, a lot of the B cells develop in the bone marrow. But I want you to think of them in another light, okay? B lymphocytes basically are involved in antibodies. That's the B of B cells. And T of T cells basically is involved in touch, because to activate a lymphocytes, whether it is an effector cell to directly one-on-one kill the organism, or whether it is a regulatory cell, the CD8 or the CD4, they have to require a surface contact or touch. And of course, plasma cells are B lymphocytes that have developed or differentiated further to making large amounts of antibodies stored in the Golgi apparatus. That's why the cytoplasm of a plasma cell is abundant and clear. It's basically a storage factory for antibodies. And of course, just as equally important as the T cells are the macrophages. The pathologists always call these cells histiocytes, but you know, they should get with the program and call them what everybody else does, the macrophages. And probably even a better name for this lineage of cells are APCs, because besides phaging or engulfing or actually having the capacity to swallow up, phagocytize particles and cells, and even sometimes your own cells are also involved in apoptosis. They basically have the contact with the offending antigen, and they present them to the T cells. So I don't like the word histiocyte, although I used it my whole life. Macrophage is the common word, but this whole lineage of cells is probably best termed antigen presenting cells, or APCs. One of the APCs, or one of the macrophages, is a dendritic cell. And think about it. If a macrophage's job is to make surface contact with a unrecognizable or untolerated antigen, whether it's a self-antigen or whether it's a bug, it has to have a lot of surface contact, wouldn't it? So they're often amoeboid. And imagine a cell that looks like a guy with a spiked haircut, you know, stars going out, little arm. Well, that's maximum surface contact. And those are why they call dendritic cells, because they look like dendritic processes coming out of neurons. And then we have this family of cells, still kind of mysterious, which you might call natural killer cells. If you want to think of them as basically lymphocytes having more of a function of a T lymphocyte direct killer cell, you can. I know they're classified separately from the T cells, but I've been told that they ultimately all come from the T cell line. So you want to make it simple? two kinds of immune cells, lymphocytes and macrophages. Well, what does a lymphocyte look like? Well, this is what it's going to look like in your office, because it's a regular blood smear. It has a round nucleus. It has a little lip of blue cytoplasm. And they talk about the diameter of this spherical cell being 10 microns, which you can see just is a little bit bigger than the red cell. So the measuring stick of the blood film is the small 10 micron spherical lymphocyte. Here you have some lymphocytes mixed in with platelets and blood cells. And look, even though it looks like they're smooth, even on this SEM, you can see there's tiny little hairs. Look at, and even on another SEM, and most lymphocytes are very, very hairy. Okay, I wouldn't call them dendritic cells, but they all have these little microvilli processes. And here is a lymphocyte, which is what we call an atypical lymphocyte or activated lymphocyte. Very often you see these when they come in contact with viruses and viral infections. This one happens to be from mono, which is a viral disease. And that's why sometimes the lymphocytes lose their totally spherical shape and they get a little non-spherical, and that's why they could be mistaken for monocytes. And that's why the word mononucleosis is not a proliferation of monocytes, but a proliferation of atypical virally damaged lymphocytes, or we'll say virally activated lymphocytes. This is what a lymphocyte looks like on transmission electron microscopy. And just like you were told with the plasma cells, the chromatin is often the densest towards the periphery of the nucleus. That's true. Plus you can see these little microvilli again. But the main place you're going to see lymphocytes in our labs are under regular light microscopy. Now think about it. If you have cells that are basically spherical, but they have just a little bit of cytoplasm, it's going to look like a very cellular blue infiltrate. So when you show somebody like a pathologist, or you, after you've seen a few of these labs, clusters of cells like this in the human body, you're going to automatically know that these are lymphocytes without even having to go under really high power on the microscope. But look, some of these lymphocytes are mixed in with cells that have a much bigger amount of cytoplasm. And these chronic inflammatory cell infiltrates, whether they're lymphoid or part of a chronic inflammatory process, have both the mononucleated cells of the lymphocytes and the mononucleated cells of the cells which often accompany them because they have to, which are the macrophages. So maybe I should say that again. When you see cells that are like this, you could probably instantly assume they're lymphocytes. If they're mixed in with cells that have more cytoplasm, but still a rather round nucleus, then you could probably call those macrophages. Okay. Let's talk about lymphocytes a little more. Actually, about 1% of all cells in the body, but we still have about 2 trillion of them. So think about that. If you have 2 trillion lymphocytes and it's only 1% of the cells in the body, then you probably have, what, 200 trillion cells in your body? Is that possible? Well, it's probably the second most common type of cell, but it's only 1/10 as common as the mature red blood cell, okay? And even though you have more neutrophils than lymphocytes, usually on your peripheral smear, you generally have about 10 times more lymphocytes in your body than you do neutrophils. Here's a cool picture of a macrophage, and we said that they have to maximize their surface area. This may not be a dendritic cell, which has these little cytoplasmic processes coming out of everywhere, but they do have these little amoeboid things, and you could almost think that this cell has a brain because it's going after the surface antigens of this bacteria here, okay? So macrophages make the contact with the antigens or the microbial antigens, and then they present those to the T lymphocytes as part of a scheme of immunity. Here's a macrophages within a lymphocytic infiltrate. Remember, the nucleus is still kind of round, but there's abundant cytoplasm. And in almost all macrophages, you see a lot of granules because they're rich in lysosomes to digest and kill a lot of cells and cell products. But they also take up pigments, okay, whether they're external, extrinsic pigments like tattoo pigments or perhaps anthracotic pigments, and they also chew up pigments in the body like blood pigment or sometimes bile or sometimes even melanin. So these are the macrophages. They phage as one of their main topics, or phagocytosis, and they present antigens, even more importantly, to the T cells. Here's another macrophage. Now, I know we told you that they basically have a round nucleus, not spherical, but potato-shaped, or what they call vesicular or round. But you can see that they're a little bit convoluted, but they still have the chromatin towards the surface. So even though we call them macrophages, derived from monocytes, it's really not a perfectly spherical nucleus like a lymphocyte. So, you want to know a general algorithm? If you see any cell mixed in with lymphocytes in Path Lab, and we will, it'll be easy to recognize the lymphocytes right away because they look like they're all nuclei. But the macrophage will have abundant cytoplasm, and it's very often you're going to either see, even on light microscopy, a granularity configuration to the cytoplasm. Here is the type of lymphocyte, which is differentiated. It still has the chromatin at the periphery, but now it's starting to make abundant antibodies, which are stored in this clear area, which they call a HOF or a Golgi apparatus. And if you see a cell anywhere that has a round nucleus, but an oval cytoplasm with a little clear zone in the lip or the prominence part of the cytoplasm, and the chromatin is peripheral, then that is a plasma cell. I can't think of any other cell it could possibly be. But even if it has only 3 rather than 4 of these classical characteristics, it could very easily be a plasma cell anyway. Here's a macrophage that has really maximized its surface area for antigen touching and searching. And this is called the dendritic macrophage, or just a dendritic cell. And they go by various names. They're in many places. They're in the liver. They're in the brain. They have other names. In the liver, they're called Langhans cells. In the brain, they're called microglia. But they're all part of the macrophage antigen-presenting cell system. Here's a natural killer cell. And what is it doing? It looks like it's killing stuff. Okay? Well, natural killer cells, basically, they come into contact with bad cells, okay? And often just directly kill them in kind of a similar fashion to the so-called killer or effector T cells. They have an enzyme called granzyme, okay, and they basically send cells and even normal cells in apoptosis, but also damaged cells and necrotic material in bugs into digestion factory called their granular lysosomes. Okay, a natural killer cell. I think, there was a joke by Rodney Dangerfield, who was a famous old comedian, and he says that his football team was so rough that whenever it came in contact with the quarterback, it then just kept running and went after the quarterback's family too. I mean, that sounds funny, but a natural killer cell is really a natural killer cell. In fact, the rule is a natural killer cell will kill everything unless the cell has a way to turn off or activate its inhibitory receptor. And if you are a normal cell, you have that capacity. If you are not a normal cell or you lost that capacity, the natural killer cell will kill you. So here we go. And when the inhibitory receptor is engaged or activated, the natural killer cell leaves you alone. It doesn't attack you. It doesn't sack you. It doesn't kill your family. But when the cell does not have the capacity because it's not recognized ultimately by the MHC system, or it's lost its capacity, then the natural killer cell will kill you. And that's not really, doesn't sound directly like an adaptive kind of an immunity, does it? So I don't know, you might think of a natural killer cell as being part of innate rather than adaptive, but of course it depends on recognizing the antigens present on either normal cells or virally infected cells, let's say. Okay, so here's the general scheme again. You have your APCs, macrophages, or dendritic cells, basically come in contact. They are now presenting the material, the antigens to the T cells, which are the controlling, the smart cell. And there's two kinds of smart T cells. There are the regulators, which are called CD4, and they're generally smarter than the effectors, which are CD8, because they are the ones that are then thinking about, let's start generating cytokines to control this whole immune process. And then there's the CD8 cells, which are the direct soldiers, much in the same way the natural killer cells are. And sometimes, you might have a hard time thinking, well, what's really the difference between a CD8 and an NK, because they are both direct killers. But remember, they kill in a different way, and the NK cells probably are more likely to kill anything. These are a little bit smarter, okay? The B cells are the lymphocytes, which ultimately might differentiate the plasma cells, although B cells in themselves have immunoglobulin on its surface, and that's one of the way of detecting them, okay, and classifying them as well. So here you go. Here's your B lymphocyte ultimately producing or generating the plasma cells to coat the antibodies onto the microbes. And then you can have the complement system kicking in and antibodies coating this to make it even more adaptive or more likely to be phagocytized, what they call opsonization. And then you have your CD4 lymphocytes after being presented with antigens, generating cytokines, which are going to either start the inflammatory process or, okay, hold on, I have to. Then you're going to have the cytokines being generated to either stimulate the B lymphocytes for antibody production or get this whole inflammatory process rolling or activate the macrophages. And finally, you have the CD8, which are the effectors, ultimately going hand to hand in killing the infected cell. And remember, it may not even be an infected cell. It may be a normal body cell that has lost its capacity to be recognized, and that is called autoimmune disease. We talked about cytokines several times. They generally are things generated by lymphocytes, as well as technically other cells, which basically get the process rolling, like innate immunity. Interleukin-1 and TNF or TNF-alpha are the main ones that not only are the main cytokines, but they are the main controlling cytokines. Cytokines regulate cell growth, especially lymphocyte growth, B cell growth. They activate and control this whole process that we call the inflammatory process. And they also stimulate the production of blood cells. For example, colony stimulating factor is a cytokine, and basically that is very effective in many ways at getting the white cells produced and differentiated in the bone marrow. Okay. Don't confuse cytokines with chemokines. We talked about the roles of cytokines, and chemokines are a special type of cytokine, and the only thing they do is produce attractants for neutrophils or polys or PMNs in the acute inflammatory process. So I know a lot of times that people confuse those. So basically a cytokine is anything produced by a lymphocyte or macrophage, let's say, which regulates the process of immunity, inflammation, acute, chronic. and of which the two most important regulating cytokines are TNF-alpha, interleukin-1, and chemokines are even smaller molecules, and all they do is attract polys. Let's go back to this MHC again because it's so important. We've got to discuss it in more detail. We told you MHC is a complex. It's a part of chromosome 6, the short arm, and it's in not only the short arm, but a band of the short arm, and then a region within the band, which can be, labeled by a certain name. And its whole job in the development of the immune system is to make sure that the surface antigens that are present on cells are recognized or tolerated by the immune system. And it also has another part where it makes complement, because if it's not tolerated, then that whole family of complement proteins is going to form a cascade and ultimately destroy the unrecognized cell or unrecognized antigen, or cell with unrecognized antigens. Here it is. Here's a chromosome. There's a centromere. There's the long arm. It's called Q. There's the short arm. It's called P. And in the 6P, the short arm, the 21 band, and then the 0.3 region, they number them as you go away from the centromere. You got 3 areas. One of them is called 1, one of them is called 2, and one of them is called 3. And it's not 1, 2, 3 as easy as this. It's actually 2, 3, 1. But in area 1, you're making antigens for all nucleated cells, including platelets as well. In number 2, you're making the things that recognize these antigens, like, antigen-presenting cells, macrophages, even some lymphocytes, okay? In number 3, you have your cascade, your whole battery of complement system proteins, which, if the cells are not recognized, it then kicks in the complement system to break and lyse and kill that cell. It may not. You may have cells that are recognized or not tolerated or not recognized, but the complement system may not be kicked in. But if it's kicked in, the cell is toast. It's history, okay? It's lysed. The membrane is lysed. So now that we had a big overview in this first half hour of the so-called immune system, this is basically going to be the outline or the rest of what I'll be talking about, okay? You might say, well, now that we know how everything should work, what can go wrong? So here are several ways things can go wrong. One of them is to have a hypersensitivity reaction. These are just names of abnormalities that could develop, and basically they always talk about 4 different types, okay? The other is the concept of failure of immune recognition, or failure of self-tolerance to your own cells. And those are called the autoimmune diseases, and they create a chronic inflammatory process, and they're basically diseases in which your body doesn't recognize your own body. And of course, we have the immune deficiency syndromes as well, whereas cells, especially lymphocytes, do not develop properly when you're born because of genetic problems. And these are the primary or genetic immunodeficiency syndromes. And then you have your secondary or acquired, or the ones you're not born with, immunodeficiency syndrome. So what happens if we say we have an acquired immunodeficiency syndrome? Hey, we have something called AIDS. But technically, if you're born with a problem because of genetics in the development of your immune system, particularly the T and B cell differentiation, you then have a genetic. So you have AIDS and GIDS. So we're going to talk about the primary or genetic ones, and then we're going to talk about the main acquired immunodeficiency state. But remember, anything that could happen to you in your life, okay, even vascular problems could interfere with your ability to form a proper immunologic reaction. Of course, AIDS is the one that we all say is the main one. So what are the 4 hypersensitivity reactions? Well, we could make them real simple at first and then go into them in more detail. Type 1 is immediate hypersensitivity. Boom! Like an allergic reaction, or a rash, or hives, or they call them urticaria, or even like a severe acute allergic reaction where you may die from an overall acute severe reaction called anaphylaxis, okay? Or you may have an antibody-mediated hypersensitivity where you have antibodies coating your cells. Now, if these antibodies fix complement, the cell will die. If the antibodies don't fix complement, they still may damage the cells as well, okay? And then you have your immune complex-mediated hypersensitivity. That's when antigens and antibodies are now just circulating throughout their system. So let's say that you have a lot of antigen-antibody complements right now circulating throughout your system. Well, what happens to them? Well, they could stay in the blood, but the 3 most likely places that they'll be deposited are either your endothelial cells, the dermal epidermal junction of your skin, your synovium, or your glomerular basement membrane. Actually, I said 3, but I rattled off 4. But the main magnets or traps or filters for antigen-antibody immune complexes are your endothelial cells, your glomerular basement membrane, and your synovium. And many arthritises, many glomerulonephropathies, okay, many vasculopathies or vascular diseases are all because of the deposition of these immune complexes. Now, in cell-mediated immunity, we're not concerned with antibodies coating antigens and floating around, or antibodies coating cells. We're concerned with cells now. It's cell-mediated. And the primary cell in cell-mediated immunity is the macrophage, because in cell-mediated immunity, we now have direct cellular contact between macrophages and antigens, often in a cluster. So many of the macrophage cluster inflammatory reactions, which we call granulomas, are classical examples of cell-mediated immunity, of which the TB test, as well as TB granulomas, are one of them, or any granulomatous disease technically. So here's number 1. Immediate means seconds in immediate hypersensitivity. You get a shot of something you're allergic to, boom. You're exposed to something and roaming through the woods and you get immediate rashes or itchiness, that's immediate. Basically what happens is that the antigens trigger off your mast cells to degranulate, to release histamine, and now you have dilatation of blood vessels and other substances which not only dilate your blood vessels, but constrict the smooth muscle of your bronchi, okay? And the late hypersensitivity is not immediate or like seconds or minutes. It's usually in the terms of hours or days, but basically it's the same sort of process. But in hours or days now, you're more likely to have an inflammatory cell reaction. In immediate hypersensitivity, it's generally too quick to call in all of the neutrophils. In type 2, we have antibodies attaching to a cell surface. There's a concept called opsonization, where antibodies are coating the cells, and in the process of those antibodies coating the cells, they make that cell more likely or more ready or more amenable to being phagocytized. But when the antibodies coat the cells, they may also kick in the complement system of these, a whole wide variety of complement protein components, of which C3, of course, is the final and last one, ultimately resulting in the rupture or the lysis of that cell's membrane. In number, oh, here we go. Here's some type 2 diseases. Whoops. Autoimmune hemolytic anemia, antibodies coating your red cells. Idiopathic thrombocytopenic purpura, antibodies coating your platelets. Goodpasture syndrome, antibodies coating the basement membrane of your kidneys and of your lung, okay? Rheumatic fever, basically antibodies being sensitized against portions of your body that were generated by previous exposure to a strep bacteria. How do you like that? It's kind of like the bridge between infectious disease and autoimmune disease, because we said your antibodies can react directly against bugs, or they could react directly against yourself. But what if they react against a bug, and then a few weeks later, those same antigens react directly against your body after the bug is gone? Well, that's a concept of what you see in rheumatic fever and a few other diseases too. Myasthenia gravis, antibodies reacting against your own neuromuscular junctions. Graves' disease, antibodies reacting against your own thyroid, okay? The receptors, the TSH receptors, switching them on, okay? Pernicious anemia, antibodies reacting against intrinsic factor, made by your parietal cells. You get anti-parietal cells antibodies, anti-intrinsic factor antibodies. Here's type 3, and we have circulating antigen-antibody complexes. We said these are 3 or 4 places where they're most likely be trapped up. And many autoimmune diseases, you'll find immune complexes either freely circulating or depositing in places like blood vessels, glomerular basement membrane, floating around in general, or technically against not only DNA or various types of DNA, but the proteins that support DNA, the histones, okay? That's basically the problem with systemic lupus erythematosus, which is the mother of all systemic autoimmune diseases. I might mention that there's something called a Arthus reaction, which is a general feeling that occurs like in hours or days. And then there's a systemic type of reaction, which is called, let's get the right name for it. A serum sickness, okay, which basically occurs later than the Arthus reaction. But it's the same process, being sick, having symptoms on the basis of circulating immune complexes. How do you like that? Here we go. There's type 4. Tuberculin skin reaction basically simulates granuloma formation because TB does. So when you use TB type antigens, which are non-infection, it creates these little clusters. If you look at them microscopically, they're mostly collections of macrophages, okay? So type 4 hypersensitivity is cell mediated. It's by the T cells, but it results in these T cells generating a whole bunch of cytokines, which eventually result in these clusters of macrophages, some of which are even giant cells, where the macrophages fuse into a cytoplasmic mass. The nuclei fuse into a single cytoplasmic mass, directed by these various cytokines like TNF, interleukin 2, interleukin 12, IF interferon gamma, okay? So basically that's type 4. And you're thinking, well, yeah, these are nice things, but how do they fit into a lot of the specific diseases? Well, we'll go into some of the specific diseases. There's our summary, 1, 2, 3, 4. Let's talk about another concept before we go into some diseases, because it's always a big thing. Why are transplants rejected? Well, we know why they're rejected because we just told you why they're rejected. That many of them are non-tolerated, non-recognized antigens. But clinically, we'll take the case of a kidney. A renal transplant could be rejected following 3 clinical scenarios or patterns. One of them is very, very acutely, and that could even be within minutes, okay, or hours. And that means that something has really gone wrong, and your antibodies are reacting against the graft. And the first place they react is in the endothelium of the donor, okay? So in hyperacute, and that may take only minutes, and sometimes some of the surgeons that do the transplants have told me that if it's going to be a hyperacute reaction, they often know as soon as they open up the clamps that that organ is not being perfused, simply because the antibodies of the host are reacting against the endothelium of the donor. Okay, there's not enough time to have an infiltrate of cells. But if we extend that time now from the hyperacute into the acute, that can go on between days and months. Remember, this is not the type of acute inflammation we see with neutrophils. These are basically cellular T-cell infiltrates, and they can appear anywhere from days to months. It's still called an acute renal transplant rejection. So if you see a kidney that is now infiltrated with lymphocytes, even though it's lymphocytes, we can still call it acute rejection. If it's in the time period of days to months, that's an acute pattern. And of course, the chronic pattern, now we're talking months, perhaps even years. The chief component of chronic inflammation, long-term, would be fibrosis. So in a renal transplant that has been rejected, perhaps after months or years, you now slow vascular fibrosis, okay? Not only vascular, but everywhere else in the kidney as well, because that's the feature of chronic smoldering inflammation. I wanted to say something else about this. Maybe I'll remember it. Here's hyperacute. Look at that vascular endothelium. It's damaged. But in the acute, you have infiltrative cells, T cells, these are T cell markers. And in chronic now, not only do you have fibrosis within the blood vessels, but outside of them as well. Okay, now we're into a big topic, and we're about half done, believe it or not. So I know I've thrown a lot of stuff at you, and maybe I've been talking a little bit fast, and I hope I was not talking too fast. So I'm going to take a nice break, and most of the stuff we talk about in the second half will be fairly much more clinical, but it will be applications of all of the processes that we talked about in the first half. So I have done a special type of music for today for our halftime break. Usually I have more rock and roll, by Chuck Berry, but I thought today I would take the most sacred songs of the major religions of the world. Islam, Christianity, Hindu, Hebrew, and I asked people around, what is the most sacred song of Islam or of Hinduism or Judaism? I already know what the most sacred song is of Christianity because I am one, but we're going to play examples of those during our break. And even though I don't understand them, they're all in languages which are not English, I think you could feel the magic and the inspiration and sometimes even the joy